This is the second session on endocrine disrupting chemicals. We've already seen that our lifestyle, we're exposed to all kinds of chemicals from the outside world, novel chemicals the body's never seen before, but that occurs throughout our lives. Even eggs and sperm, before they even make us, are already being exposed. The mitochondria within the eggs are being exposed. Throughout our pregnancy, we're exposed to not just exogenous chemicals, but to changes in nutrition and nutritional components, to stress. And the question is, to what extent do they affect us as we grow up? To what extent do they affect our health and our development and our susceptibility to disease? This shows the human population over the last 10,000 years or so, and we've already seen how it's shooting up enormously over the last 200 years, coinciding with the Industrial Revolution, where we've utilised power stored over hundreds and thousands of years in fossil fuels, and the Agricultural Revolution, where we've utilised novel pesticides and fungicides. And we've, over that time, introduced, especially in the last 50 or 100 years, novel pharmaceuticals. Some of these, the pesticides, fungicides, pharmaceuticals, are targeting biological systems anyway. The use of things like plastics aren't targeting anything specifically, they're just useful, but they incorporate many chemicals which may affect biological systems. Many of the chemicals are actually very persistent. They're not broken down very easily. They're not broken down within the body, and they will bioaccumulate. Here we see small fish, and as they absorb some of the chemicals, they will take them in, it will go into the fat, and as they grow up, they eat more and more, they absorb more and more, and the levels within the fish get higher and higher. That's bioaccumulation. On top of that, things will biomagnify up the food chain. Here we have krill, eaten by fish, eaten by seals, eaten by polar bears. And you have huge increases in these persistent chemicals as you go up the food chain. The bioaccumulation and biomagnification of substances is well illustrated if we look in the Arctic. Here, persistent chemicals accumulate in the snow. When the snow melts, they go into the water, and then they're free to work their way up the food chain through the aquatic animals, fish, and up to the whales and the gulls and birds at the top of the food chain. Remember, this is a log scale, massive bioaccumulation as you go up the food chain. It was in the early 1990s that the story of endocrine disrupting chemicals really took off. It was because of observations from various parts of the world which all combined together to suggest there was a real problem with some of these chemicals. There were intersex fish observed in rivers in Britain, abnormal sexual development observed in lakes in Florida, oestrogens coming out of cell plastics in scientific labs, and a general concern about what sperm counts were doing in the human population. The early worry was that all these chemicals are estrogenic. The focus on estrogens really came from the early studies on breast cancer. Breast cancer, which was very prevalent cancer. There'd been a lot of work on it, and it was known that many breast cancers were driven by estrogens. Because of that, there was a lot of research done on estrogens and ways of measuring estrogens and estrogenic activity. So there were very sensitive assays for measuring anything estrogenic, and there were a lot of studies on estrogen receptors. So the technology was there, very sensitive systems to detect estrogenic activity. Let's have a look at some of these things. Let's start off with estrogens in cell culture plastics. It was in the early 1990s that the study of breast cancer cells in culture revealed that some culture flasks seemed to have some problems. Breast cancer cells are very sensitive to estrogens, and it seemed as though they were being stimulated by something coming from the flask. What could it be? Well, what was revealed was that, in fact, when these flasks were autoclaved to sterilize them, they released something called bisphenol A, which we now know is very prevalent in many things we use, and this was estrogenic. And that led to a greater concern. Polycarbonates are used very widely. They're used in things like baby bottles. Baby bottles are sterilized, or can be sterilized, by boiling them. If you boil up polycarbonate flasks, they will release bisphenol A. So we have the possibility that babies were being exposed to estrogenic materials early on in life from baby bottles. And this eventually led in 2010 to a widespread ban on plastic feeding bottles releasing bisphenol A from use with of feeding babies. The problem is that bisphenol A is very, very useful. It's used for many different things. CDs, photographic film, although very few people use photographic film now, glasses, dental sealants, containers, small and large, pipes, including pipework which catheters, etc., in dwelling lines, taps, in medical devices, water pipes. The estimates in 2018 was that there were 6 million tonnes of bisphenol A being used per year, and that was going to increase by 6.5% each year for the next 7 years. Let's go on. What about intersex fish? How do they throw light on the issue? Scientists at Brunel University, northwest of London, identified a peculiar problem. There was a high incidence of intersex fish in fish living downstream from sewage treatment works. Upstream, there was a low incidence, so it looked as though something was coming out of the treatment works which was causing intersex fish. Intersex fish were very easily identified because the testes of a normal male looked like this, whereas intersex fish, the males had oocytes within their testes. It was known that estrogens could cause sex reversal in fish, so the possibility was that something coming from the sewage treatment works was estrogenic and causing intersex fish. What could it be? Fish were actually a very good model for identifying estrogenic substances in water which might be affecting fish. In the normal female fish, the ovary, using estrogens, stimulates the liver to produce an egg protein which goes back to the ovary, accumulates in the eggs, which are then laid. But the liver responds to anything estrogenic, not just estradiol. So in a male fish, vitellogenin, if you find it in the blood, it reflects the fact that a male fish's liver is being exposed to something estrogenic. And here we can see the results of a typical experiment, housing male fish in various concentrations of sewage effluent, 25%, 50%, 75%, or 100% sewage effluent, against the background levels that you get in the fish. We can see that as you increase the amount of sewage effluent, the concentration of the sewage effluent, so the plasma vitellogenin concentration goes up massively. This is a log scale, remember. They identified a couple of substances which were weakly estrogenic. These were breakdown products of surfactants. Surfactants are widely used in industry as surface tension altering materials in industrial processes, manufacture of paints, and there was a widespread wash down the drain essentially. That's why you could see foams coming very prevalent downstream of weirs. Now, these surfactants broke down into non-alphenol and octalphenol, and these things were weakly estrogenic, and there was a high concentration of them in sewage, in polluted rivers, unpolluted areas. The concentrations were much lower. Observations were beginning to accumulate. There were the alkyl phenols, the breakdown products of the surfactants, seemed to be weakly estrogenic. Pesticides, often very persistent pesticides, things like DDT, were also weakly estrogenic. PCBs, which were very widely used in industry but now banned. Phthalates, widely used for changing the properties of plastics. Bisphenol A, widely used again in everything from food can lacquers through to dentistry, through to the water pipes. All of these seem to be weakly estrogenic as detected by those very sensitive breast cancer cell assays. But look at them, there's something peculiar. This is them structurally, but while they're weakly estrogenic, none of them actually seem to resemble estradiol very much at all. So what's going on? Some of these observations were certainly scientifically interesting. The intersex fish, the DDT metabolites which cause the abnormal sexual development in alligators, estrogens coming from various plastic materials. But what really set the whole story alight was a widespread public awareness that things might be actually affecting human reproduction. Was there a decline in sperm counts going on in the human race? The issue about sperm really began in 1992 with a paper by Carlson et al., who published in a British medical journal. They looked at 61 publications which had been published up to 1990, and for each publication they looked at the sperm counts which had been reported. So here was a paper in the early 1940s who said that human sperm count was somewhere around 60 million per mil. This one in the late 1970s was saying, oh no, it was somewhere about 130 million per mil. So each of these dots represents a different paper, and the size of each dot represents a number of people, men who had been sampled in that paper. So big dot, lots of people, small dot, few people. And they said if we plot all this data together, it looks as though sperm counts have been dropping consistently over that time. The trouble is that this has got some flaws in it. Is it really on to draw a line like that with these data points? It looks as though the data is heavily biased from the data up here compared to the rather few data points down here. The reason why there are lots more data points up here is because of assisted conception. The technology to come in, Louise Brown was born in 1978, so assisted reproduction in IVF really was taking off at this time. So many more studies of sperm counts, relatively few in the 50s and 60s and 1940s, because to be honest, nothing much could be done about male fertility or infertility. Another thing is technology had changed. Back here in the early 1940s, 50s, then everything was done by looking down the microscope and counting each sperm by hand. Up here, computer-assisted technology was coming in. So there was a question mark about whether this data was really real or not. But sperm counts are remarkably variable. This is looking at the sperm counts of a single man over a 4-year period, 1990 to 1993, with regular visits to the clinic to assess his sperm counts. And you can see they go up and down, up and down, up and down, up and down, from less than 55 million per mil up to more than 240 million per mil. Very variable. So they're not a good indication. Certainly a single sperm count is not a very good indication of what that man can really do. And sperm counts show a seasonal variation. Here are sperm counts in 4 European cities in the winter months, the yellow, and in the summer months. And they're consistently lower in the summer. So what's going on there? The variability in sperm counts was a real problem. Were sperm counts falling or not? Some studies said yes, some studies said no. So this is a report on the BBC website in 2012. French sperm counts fall by a third, it said, and that's after a paper came out of studying more than 26,000 French men, and the number of sperm seemed to be going down. But when this was put to one of the UK's specialists, he commented, "I'm left questioning how much of the changes described in this paper are simply a function of alterations to lab method. I would urge much caution in its interpretation, as there remain too many unknowns. In my view, the paper certainly does not resolve the issue of whether sperm counts have declined or not." Much more extensive data came out in 2017, where a very large study tried to re-examine this whole problem. Essentially, they reported on a much larger set of papers from all over the world, including fertile and infertile people, a very large number. And what they found was that there was seemed to be a gradual reduction in sperm counts from the mid-1970s down to 2010. but that because of the large number of samples, they could do a much better analysis. And they said declines were only significant in studies from North America, Europe and Australasia. While sperm counts have the disadvantage of being very variable anyway, there was other data coming through to suggest that other changes in male reproductive health were occurring. Testicular cancer, which is very easily detected, recorded, you can't miss it. The rates of testicular cancer have been going up throughout the world over the last 30-40 years. This is data from Europe, looking at Central and Eastern Europe, Northern Europe, Southern Europe, Western Europe. This is a log scale, and you can see the rates have been going up very markedly. What's going on? The issue is well illustrated from the very good studies which have come out of Finland and Denmark. These are two Scandinavian countries, and they show rates of testicular cancer, both of which have been increasing, but between the two countries, Denmark and Finland, the rates are very different, much lower in Finland. And it wasn't just testicular cancer. A whole range of things seem to be different between the two countries. The Danes had smaller testes, a high incidence of undescended testes, hyperspadias. Hyperspadias is where the urethra doesn't end at the tip of the penis, but instead of that, somewhere up the shaft. And the Danes had poor semen quality. So this range of male reproductive characteristics, testes size, undescended testes, hyperspadias, semen quality, was given the term testicular dysgenesis syndrome. And the rates of increase, as illustrated by testicular cancer, was much higher than would be expected from some kind of genetic basis, which seemed to be almost certainly due to some kind of environmental cause and maybe endocrine disrupting chemicals. Adult men with testicular dysgenesis syndrome disorders often exhibit problems in their testes, histological problems in their testes. This looks like a normal tubule with lots of sperm in it, but many men would show these, Sertoli cell only tubules. No sperm are being produced by these. These sets of observations highlight the likelihood is that we're looking at developmental problems. Testicular cancer may well be initiated, or the likelihood of getting testicular cancer may well be initiated early on in life. Certainly, where the urethra doesn't end at the tip of the penis, that's a developmental problem. The rate of undescended testes is a developmental problem. So, early on, something is happening to males. To understand this set of problems in humans, we really need an amenable experimental model. Rodents are often used in experimental studies for a whole variety of reasons, but for reproductive studies they're particularly useful. There's a short generation time. Pregnancy is only 21 days, lactation 2 or 3 weeks, and for mice they can become pregnant again by the age of 35 or 40. They produce multiple offspring, both males and females in the same litter, and each female has 2 uterine horns, so if necessary work can be done on fetuses in one horn and the other horn can serve as a control. This is a typical type of experiment which could be done. You take a rat, mate her so she becomes pregnant, then expose her to any particular endocrine disrupting compound for different concentrations for different lengths of time. This one is dibutyl phthalate, and look at the outcome in the offspring. And in the early days it was obvious that such interventions could produce male offspring with a variety of reproductive disorders, like cryptorchidism, malformed external genitalia, reduced sperm count, changes in the anogenital distance, all of which were characteristic of a disruption of androgen action. And all of these resembled human testicular dysgenesis syndrome. So here is a typical animal model which we could use to investigate what was going on in humans. So it looked as though the scene was set. We had the problems, we had experimental tools to look at the problems, but in trying to unravel the answers, there were a whole host of confounding variables, and we'll start looking at those in the next section.